everyone. It is an honor and privilege to introduce our today's guest, Dr. Prashant Kharkar. He is Professor of Medicinal Chemistry from Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. He is working as Dean Student and Alumni Affairs, and he has a startup in his name, that is Minoni M Life Sciences Private Limited, which is situated in Thane, and it focuses on biomimicry based therapeutics. He is a drug discovery scientist with rich experience and expertise in medicinal chemistry, computer-aided molecular design, cheminformatics, biomedical sciences with more than 15 years of research experience. He has recently completed preclinical development of cancer stemness inhibitor-led molecule, which is investigational new drug, and it is application approved, and phase 1 studies on solid tumor bearing patient are in progress. He has also completed application for first-in-class viral entry inhibitor, which is for COVID-19 approved. Phase 1 clinical trials in healthy human volunteers are in progress. He is presently an inventor on 25 national and international patent applications, and he has guided 5 PhDs and more than 25 PG students so far, and currently 9 PhD students are registered under him. He has published 90 research papers in reputed international journals, and he is the recipient of more than 8.5 CR grant from various national funding agencies and industries. He is editorial board member in Annals of Oncology Research, Springer, and Nature Discovery Biotechnology. So we have an eminent personality with us today. So without taking much time, I would like to invite Sir to please start with the session.
Okay. A very good morning, I would say. I mean, no matter what time of the day you meet, if you're meeting for the first time, even if it is like 12 o'clock in the night, you say good morning. So, good morning to everyone. I can't be more happy to see all the enthusiasm with all the Sakis, like Madam mentioned at the beginning. There is so much positivity in the room, and I salute to everyone here for all the work that you are doing for the betterment of the society. And I would definitely would like to be part of your efforts in some way. Thank you so much for inviting me here. And I can't be more happy again to see the hall full, because in today's times, getting students to come and sit for a lecture and the hall full, this is at times a rare scene. But believe me, I'm so very happy, and I'll be very happy to talk about this particular topic, which is very relevant. And then I've been practicing this particular field. Of course, AI is relatively new, but then the I part of it, intelligence in drug designing. So I have been doing that for quite some time because it was my childhood dream that I wanted to discover a drug. I did not know how a drug was discovered, and it just happened to be it just like when you want something very desperately, the universe makes it happen, and this is exactly what happened. I wanted to discover a drug, and finally I landed up in ICT. Of course, coming to ICT was very, very difficult unless you are all India topper in GATE after your masters. You can't really be in ICT. So I worked very hard, but then all you can do is work hard, and then you can't really decide whether you are going to be topper in a national level exam. But the only thing you can do is you work hard, and the universe listened to my prayers, my hard work. It responded. I stood first in GATE 1998 in pharmacy, all India level exam. And then I ended up with a person, Professor V.M. Kulkarni, who was the then father of molecular modeling in India. And that's exactly where I continued my journey. He was to retire in less than 3 years after my master's, and then I persuaded him to, take me for his for PhD. I was his last PhD student, and again to pursue my dream that I wanted to discover a drug. But then, as I got acquainted with how difficult to discover a drug is, and it's like no one man's job. There are multiple people who are involved. In fact, whatever we do in basic sciences, ultimately it has some or the other connection into drug discovery, the ultimate application, because we humans, we want drugs for multiple ailments, and everyone has experienced in pandemic what happened. The vaccines were approved without much of trials, and then a lot of issues have people are still facing a lot of issues. A lot of people have died supposedly because of vaccine adverse effects. But anyways, I'll be talking about this topic, that is artificial intelligence in drug designing and diagnostics, bridging molecules, models, and medicine. And before I begin, let me just thank, of course, all the Sakhis, Indian Women Scientists Association. Then, of course, Board of Research in Nuclear Sciences, is BRNSDA, for sponsoring activities of this scale and magnitude. Rayat Shikshan Sanstha's Karmovir Bhavarao Patil College, Vashi. Definitely. Thank you so much. And then, if I don't thank the scientific community, my acknowledgements are going to be incomplete. So, scientific community for everything, whatever little I know in this field, whatever I do, it all I owe it to the scientific community. So, like we have seen artificial intelligence. So first, I'm just going to spend a couple of minutes, all the three slides, all the three words that you have in the title. What is AI? Right? So AI, artificial intelligence, everyone knows about it. Everyone has their own version of AI. Like some people think that AI is kind of a machine, AI is a program, or AI is anything that does some fancy things. I'm sure most of you are using Chat GPT or any other large language models like Claude or anything for that matter, Perplexity and whatnot. So AI is machines that learn patterns from data. So all you need is data. Say like, how does AI know that? Like when we put GPS, or you tell Google Maps that I want to reach to college, so it told me 37 minutes, and then we reached with the plus minus 1 minute here and there. Exactly during that time or in that period, we reached. So how did it actually know? And this is exactly where I would like to bring, and of course, everybody is aware of it. AI is part of our daily lives. Guess how? Everyone knows about it. In fact, last night for this particular preparation, slide preparation, or presentation preparation, I was searching an example on arrhythmias, cardiac arrhythmias on diagnostic. And then just before sleeping, I was just scrolling through my Facebook. So it actually showed me some of the posts related to cardiac arrhythmias. So we are being watched, we are being heard. There are downsides to the technology, but again, we are not here to discuss that. One has to be careful, because with every technology comes responsibility, and the way we use AI or these technologies for the betterment of our lives, there are people who are also using it for creating bad activities, or are part of bad activities. But anyways, so I'm just going to bring to this particular a circle within a circle, or an oval shape within an oval shape, just to give you a brief overview. AI is the broader picture. Then comes ML, machine learning. So machine learning is a subset of AI. And then comes DL, that is deep learning. right? So which is again a subset of machine learning. Again, most of you will be aware of this. So when you say deep learning, we are talking about deep learning as the machine learning using deep neural networks. Again, in fact, it's a very interesting technology. Those who have discovered deep learning networks or neural networks, they don't know what actually goes on in the hidden layer. You have got the input layer, hidden layer, and then multiple hidden layers, and you have got the output layer. The inventors of deep learning or deep neural networks, they don't know what exactly goes on. It's just that you give a good data, it gives you a good outcome, right? So then comes machine learning, which is a science of teaching computers to learn from data without being explicitly programmed. So everything learns or everything revolves around data. If you give computer good data, it will give you good output. If you give it junk, it will give you junk output. And then the broader picture, AI, which is basically, so these are all formal definitions. AI is engineering of machines and programs that can learn, reason, and make smart decisions like humans. Like every time you use Chat GPT, that data, in any question that you ask to Chat GPT, even if sharing some emotional stuff with Chat GPT and it is responding to you, so it's learning. It's learning without being explicitly told, and this is exactly where the brighter side as well as the downside. In fact, I think yesterday only I read some news where some users, they were trying to use some abusive language and then trying to tell Chat GPT that it's okay to use this abusive language. So it wouldn't think, it wouldn't know that it's okay to use the abusive language, right? Otherwise, it will be very nice to you. You say hello, it will say hello to you, and all those things, right? So again, the downside of the technology. So there are people who will be on that other side. But anyways, so in science, now because we are talking about the application of AI in science, particularly drug designing and diagnostic. So in science, AI learns patterns in molecules, diseases, images, and genes, right? So if you look at, I'm sure most of you are biotechnology students, right? So you all know the central dogma of life, and then how the DNA to RNA to protein and the life goes on, right? So everything revolves around the molecules, and then diseases, images, and genes. We'll see that in one particular, some particular time during this presentation. The next word is drug designing. In fact, if you ask me, I don't know, hate is the correct word. I hate to call it as drug design because my boss, my mentor used to say, you cannot design drugs, you can design molecules. Whether it becomes a drug, even God doesn't know. You have 100 molecules entering in preclinical trials, phase 1, phase 2, but nobody knows whether which will become drug or whether none of it will become drug. So molecular designing is the correct word, but I did not want to differ from the topic, so I just continued using this. So what is drug designing? So it's a scientific process, very systematic scientific process of creating new medicines that can interact with specific biological targets in the body to treat diseases. Now, I'm sure you have some idea when now this particular season is such that a lot of people are suffering from runny nose and all. So you take some medicine, cetirizine. Do you know how it acts? Probably yes, probably no. Doesn't matter, right? So you take a medicine, it gets into blood, from there it goes everywhere in the body, and in the process, it will also interact with histamine receptor. So cetirizine is histamine H2 receptor, H1 receptor blocker. It's an antihistamine, right? So the target is histamine H1 receptor. The molecule cetirizine, that tablet you take, which is actually a molecule, it goes and it binds to its target, and at the end of it, it will give you anti-allergy effect. Fantastic, right? So likewise, but then how you cannot just dream of cetirizine that this is what cetirizine structure is going to be, and it will go and bind to histamine receptor and it will give you anti-allergy effect, right? So you have to design the molecule. Of course, cetirizine is one such molecule which has come up. I mean, there are many molecules or many drugs which were discovered by serendipity. Aspirin was discovered and launched in 1896. So many years, more than 125 years, right? Such a small molecule, it is still used. And there are many drugs which were introduced like 5 years ago, but then they are not existing because there are a lot of side effects, right? So despite the fact that you design the drug so that they will interact with their intended target, give the intended therapeutic effect, but no guarantee that they will survive their lifetime. Some of the drugs like aspirin is doing so well after so long, And then rofecoxib, which was introduced like a couple of decades ago, or rather little less than that, it is now taken out of the market. There are many drugs like that, right? So what is shown here is a target, a protein target. And so there is a binding site. So whatever cavity-like structure that you see, and then in yellow ball and stick model, it's actually a molecule. This is one of the molecules that we have discovered along with Godavari Biorefineries Limited. So it's a cancer stemness inhibitor, and it is developed for treating triple negative breast cancer, which is one of the dreadful cancers which catch young women right early on in their life, and then there is not much that can be done. If it is too aggressive, you can't really do, the patient will eventually die. So we have been working on this particular aspect, like madam has mentioned. So this is one of the molecules that we have developed from scratch. In fact, we had some phytochemical lead, a natural product from some leaves, and then we have modified it systematically. It's been 13 years I'm associated with Godavari Biorefineries Limited, and this is one of the molecules that we have come up with. This has been patented. Almost 10 patents have already been granted to this and others. So what you need is, there is a target, and then there is a molecule. So there is a definitive shape, just like a lock, and you have to design a molecule, a specific key, only that key will open that lock, right? So lock and key mechanism in simple terms is what I would like to call for a drug interacting at this particular target. So remember this process of designing, and this is where the AI, I'm going to fill you in with this particular aspect of AI. Like I said, these molecules, I mean, I wouldn't say AI wasn't involved, but not a formal, it was all coming from intelligence, from the brain. There is another molecule which we have discovered, and which now the phase 1 studies are also complete. It's a viral entry inhibitor. It is not specific for SARS-CoV-2. So tomorrow there is another virus which is kind of threatening us. In that case, we believe that our molecule is likely to prevent the entry of the virus. So it's like no virus inside the body, no disease, right? So, but anyways, some other time I'll just take you through. The next part is, of course, diagnostics. Everyone understands the importance of diagnostics. In case you know somebody who is suffering from cancer, say, as common as breast cancer. So typically a small tissue will be, a biopsy will be taken, and then that will be analyzed genetically. And then depending on the genomic signatures, whether it is ER positive, PR positive, or triple negative, the course of the chemotherapy will be decided. It's very important. So diagnostic has never been so very sophisticated all these years. I mean, I would say it's a decade and a little more than that, decade and a half, I would say. There have been, like, people are talking about molecular diagnostics, where you're talking about next gen sequencing, sequencing the genome and finding out. Now, if I tell you that if you have got enough money, it's possible to know in next 15 years whether you will have a probability of getting a cancer or getting a heart attack. But the question is, do you really want to know that? Nobody would want to know, because if I know that after 15 years I'm going to have a heart attack, so my 15 years are gone. I'll be worrying about that one day, one instance where I'm going to die and nobody's going to save me. I mean, probably somebody will save me, but then it's not like when it will come, right? So you don't really want to know about something like this. The technology is available. There are powerful technologies, but then, right? So diagnostics are very important, and AI has been doing a lot in terms of diagnostics. So diagnostics being tools and methods used to detect diseases, identify health conditions, and monitor a patient's health. As simple as that. And biotechnologies in many levels are very crucial for developing newer technologies for diagnostics, for that matter. So looking backward, I mean, drug discovery is not new. People have been discovering drugs for a very long time, maybe more than 6 decades, 7 decades, long time ago. Penicillin was discovered in 1928. It came into therapy in 1939. Then isoniazid, anti-tubercular drug, came into existence or clinic in 1952. So many years. Many of these drugs were randomly designed, meaning people just randomly made molecules and they became drugs. Some of them, they are still doing well. Many of the antibiotics, they were discovered, some of them discovered serendipitously, some of them like ciprofloxacin or floxacins, they were rationally designed. Most of them, otherwise, they were serendipitously or by accidentally designed. So coming back to discovery, so what is happening today in today's world is, it takes on an average $6 to $10 billion to develop a drug. Now you can do the math. $6 to $10 billion, probably 60,000 crores in Indian rupees, assuming dollar rate is 100 rupees per dollar, or 50,000 crores. That's a lot of money. That's a lot of money. Who has that kind of money? And that's exactly where I don't know. I mean, I don't know whether, I mean, how to say it, but India has never discovered a drug. I mean, there have been instances where we claim we have discovered a drug. In fact, recently, some of the companies they have come up with drug, but none of these have become blockbuster drugs. Drugs whose annual sales are in excess of $1 billion. But then we take a great pride in saying that we have got a scientific force, very very powerful scientific force, young people like you, but still we are kind of on the back seat when it comes to discovering a drug. A lot of bright minds have contributed to development of multiple drugs. They are doing so, maybe in US, in developed nations, Indians. But then as a country, we haven't really done well. And that's exactly where it's our, I would say, it's our wish list. It should be our wish list that we discover drugs, at least for, the things that we need immediately, diseases like chikungunya and then whatnot, right? So then, because there is so much of expense involved, the need is felt that it has to be done differently. If we continue doing the same way, it's not really going to help in any way. I mean, only 5% of the drugs, 100 out of 5 drugs get the return on investment. I mean, you invest $10 billion and only 5 of the 100 drugs actually get the return on investment. Right. So you see the maths is so disappointing, and nobody would want to discover a drug. A US company wouldn't be interested in discovering a drug for some African disease. Why would they? Because there is no economic advantage. Because it's a business. Everything is about money, and that's exactly where we have to do things on our own for the diseases that bother us. And this is where we need to. So that's exactly where this is a fantastic article published in 2009. And then why I take this article is because after 2010, the AI started picking up, and then this last 15 years has been a lot of success stories have come up. And sometimes, in case it's a freely access, open access article, you can read through. Amazing article in case you are interested, right? So there was a need to streamline the drug discovery, and this is exactly where people thought maybe the computational approaches would be of greater help. And this is where, I mean, if you ask me, I came to ICT in 1990. I started working for my master's. My boss was already into molecular modeling, computer-aided molecular design. And I actually came for that purpose only, that I wanted to learn computational approaches. So I have seen the journey of last 27 years, right from 1998, where we started with 486 computers, and now we have got GPUs and supercomputers, cloud computing. I've seen all the journey, right? So it's very, very reassuring that I'm on the right path. Now, of course, I have learned, I have, whatever was the need of the time, I have worked very hard. I have learned all the new things. And now I'm actually getting into AI and amazing things we are doing. Fortunately, I got great collaborators, and currently I'm working with Earth Software for this particular aspect. So democratizing drug discovery using computational approaches. And this is exactly where the physics-based molecular modeling, where we use thermodynamics, free energy calculations, and many other things, because everything is governed by anything that happens in the nature is governed by thermodynamics or free energy changes. And that's exactly where you need to couple these physics-based machine learning or other molecular modeling techniques with DL as well as artificial intelligence. Now, again, like I told you, it takes around 12 to 15 years to bring a new medicine to the market. Now, the question is, can I bring this medicine? Now, you see, COVID, can we really wait for this particular time, 12 to 15 years? No. In fact, the fastest was AIDS drug. When AIDS came into to mankind in 1984, when it entered in human race, then the first lot of drugs were discovered in 4 to 5 years. Rationally designed drugs, HIV protease inhibitors, reverse transcriptase inhibitors, a lot of things. So that's where we can't, nobody can wait for 10 to 15 years. Maybe after that time, there won't be any disease left. There may be something else. There was no chikungunya when we were growing. Just so I know somebody near and dear ones who died of chikungunya. Is it really fatal? It's not. But then somebody dying, right? So things are going in a different way, and this is where we need to be on the vigil, working very hard, right? So this is where we need to reduce the time to discover a drug, and this is where the computational approaches and AI can help. And this is what I'm going to reiterate for that matter. Okay, so then, but then when I talk about computational approaches, is it going to happen in the air? No, I need very fast computers. They're going to be expensive for sure, but then government is doing all their bits to provide the fastest computational resources. But at the same time, I also need the methods which are advanced to suit this fast computational machines, or use these GPUs and all. But at the same time, I also need the advances in multiple different fields. I mean, like it's a heavily multidisciplinary field. I need inputs from protein structure prediction. I need inputs from computational side. I need inputs from methods, which are essentially will be used for protein structure. I think a couple of years ago, John Jumper and two other guys got Nobel Prize in for their discovery of AlphaFold, protein structure prediction. Now you see somebody getting Nobel Prize, which is the highest award any scientist can get. So you can imagine how cool it is, or how important it is when it comes to AI and the technologies that are being used. So AlphaFold has changed the scenario. I mean, there are multiple proteins whose structures we don't know. If you don't know the structure, forget about the function. I mean, merely having a sequence wouldn't really be helpful. And this is exactly where we need advances in multiple fields, without which I mean, merely having AI wouldn't really help. You also need the inputs for AI, the data, high quality data, including protein structure prediction. But at the same time, I also need chemical space, the chemical universe. When I say small molecules like aspirin, paracetamol, I need a chemical universe. What are the possible number of molecules that can be synthesized and which are drug like? The number is around 10 raised to 60, and the time lapsed since the Big Bang is 10 raised to 17 seconds. So even if the mankind started when the Big Bang happened, started making one molecule per second, we would have still covered 10 raised to 17 molecules. That's not possible at all. But then you see the problem is so magnificent scale, right? And without AI or without these high-end computational tools, nothing is possible. And this is where AI is really helping. I mean, I'll be using some technical stuff, but don't worry. The whole purpose, I cannot make you expert in AI or the drug designing or the use of AI in these fields. I really want to instigate the thought process. Even if one person is inspired from this and to take up, the career, professional career in these areas, that would be, the whole purpose will be served for, so many people taking so much hard work. Okay, so I'll just take you through some of the cool applications of AI and computational modeling in small molecule drug discovery. Like I said, I've been in this field since 1998, and I always wanted to discover a drug. And I've seen the evolution of the technologies. Not that I had access to all of these technologies, but I was fortunate to get all the tools like modeling softwares. And then we also had GPUs back in 2012, also the company I used to work for. And this is exactly where, so when you talk about designing a small molecule, so you need a target, you need its 3D structure. I mean, if you follow what happened when the SARS-CoV-2 came, everybody talked about the receptor binding domain, then it interacted with the ACE2 receptor, and then how we can disrupt this interaction so that there is no interaction, there is no SARS-CoV-2 entering inside the body, and then there is no disease, right? So there were multiple targets which came up, and this is where scientists started working on developing or designing drugs. So you definitely need a target. You also need a database of small molecules. Where are these small molecules going to come? We already have databases like PubChem, and then there are Zing database, which talks about few billion molecules, possible molecules. Not that all of these molecules are synthesized, but then this is where you need some kind of virtual database, imaginary molecules, which will still be drug-like, having a particular set of properties. And to use these few trillion molecules or few quintillion molecules, you need some kind of a computational resource. And this is where, so basically you have a target, and you remember the lock. and then you have these molecules, these small molecules are going to be different keys. So out of say a few trillion molecules, only 1 or 2 or maybe 100 keys are going to fit in that particular target, not everything. But then how do I find out that these are those 100 molecules or 10 molecules or one molecule which is going to fit into that particular target? And this is exactly where you need the tools to match the right molecule for the right query target. Right, and this is where the AI comes in picture. This is the same picture that I have shown you, a molecule bound in its active site in that particular target. So then simply you will just go to Chat GPT, and so you have no idea how it is to be done. So you say, hey GPT, tell me how can I work this out? And then the GPT answers, well, I can certainly help you. Here is the answer, and this is exactly where I'll take you through the scope of AI in small molecule drug discovery. Obviously, protein structure prediction methods. Amazing thing has done. AlphaFold 3 is released, which is free for academicians. And then there is another tool that has come up, Bolts 2, which was launched in March this year. Amazing thing. So all of these tools, you give a sequence, any big sequence, any small sequence, and it will predict the 3D structure very accurately, very correctly. And this is exactly where that is my starting point. I have a 3D structure of the protein, and then I can now basically use AI's help to take the small molecule discovery ahead. But at the same time, like I said, I need to also have a very big, large library of molecules. So there are now reinforcement learning algorithms which will generate like few billion or few trillion molecules just like that in few hours it will generate. So you tell that this is what I want, and then please generate these many number of molecules. So molecular generation, that is another scope where AI can help. But now I have got the target 3D structure. I've got few trillion molecules. I need to now make them, find them, match them, and find out which of these few trillion molecules are really going to bind to this particular target. So then I need molecules, and I need tools for compound screening. But then not that every small molecule that binds will be able to act in the body. It's like when you take cetirizine tablet, so it has to go to stomach, dissolve, get absorbed in blood, pass through liver, go to heart, to lung, to heart, and then from there it goes everywhere. So all of these things are limited by so-called ADME processes or pharmacokinetics. So not every drug, every molecule that is able to bind to the target will be able to do in the body, well in the body. And this is where you need to admit screen. It has to have certain properties without which it will not be able to reach the site of action from the site of administration. And this is exactly where you need to predict these properties and prune these molecules down. Otherwise, these molecules are going to fail in later stage clinical trials where you have already spend few $billion to develop the molecule till phase 2, and then you realize, oh my god, it doesn't do anything, it doesn't reach the site of action because in human body, because human body is so complex, right? And this is where you can't really afford to forget about these few $billion which you have already spent. So I would rather take in the early stages that a molecule which has got a high probability of success later in the clinical trials, right? So there are multiple approaches. If you talk about machine learning, supervised learning, unsupervised learning, semi-supervised learning, reinforcement learning, deep learning. I'm not really going to go in the details because that's not possible. Now you see, when you say reinforcement learning, do you remember when you started riding a bike or say cycle? Forget about bike, cycle. So did you learn on your own, or your parents or your friends, your siblings helped you? Probably your friends helped you, right? So when you start learning a cycle, initially you don't know how to balance, but then you fall once, you get injured, but then eventually you learn on by yourself. And this is what the reinforcement learning is all about. Very, very important as far as AI is concerned. So these are the broad application areas. This is one very interesting article, Artificial Intelligence in Small Molecule Drug Discovery from 2018 to 2023. Does it really work? So obviously it's a new technology. People are skeptical. And then unless there are some successful examples, nobody's going to believe. And this is exactly where articles like this, if you are new entrants, if you are curious, read these articles, right? Simply asking Chat GPT will not help you in any way. You have to actually go to the library or online. Many of these articles are available for free. You need to read this. And eventually this is another very interesting article. So again, like I said, my aim is to instigate the thought process. So some of these tools or other, some of these papers are there, which you can read. AlphaFold accelerates AI-powered drug discovery, efficient discovery of a novel CDK2, CDK20 small molecule inhibitor. Now you see CDK20 is the target, small molecule inhibitor is what they want to discover, and they have used AI for this. So this is a case study. So when you actually read about it, forget about whether you understand all the technical aspects. Probably not. Maybe you are not from that field, but you have some idea because you are from biotechnology. But then at the same time, when you read, so at least you will understand the thought process. How did they do it? What all issues they faced, right? And this is where the great learning is all about. This is another, now there are companies like Benevolent AI, which claim that they have discovered a new drug using a technology called pandaomics, using all the genomics, metabolomics, lipidomics, all those omics stuff, proteomics. So they have discovered a drug from whatever data was available for a disease state. They have discovered a new target and used their proprietary technology, say Chemistry 42 platform, and they have synthesized only few molecules. Otherwise, typically, 10,000 molecules are synthesized, and then you use them for high throughput screening or so. But they synthesized very few molecules, and within 1 year, they directly went to phase 1 trials. This is amazing. This is unheard of. This is people, nobody have thought. I mean, I've seen drug discovery for last 27 years, but then I will always be skeptical. How could someone do it? I will never believe this. 1 year, I will be out of business, right? I mean, I'm a drug discovery researcher. If somebody is claiming that with the help of AI, I'm discovering a drug in like 2 years, well, probably, but then now you see, there were a lot of skepticism. There was a lot of skepticism, but then I also read a lot. Now I tend to believe, and in fact, we are also claiming we have a paper recently, a paper out with our software, where we are saying that we really want to come up with a drug in very, very short amount of time. So then, where will this be useful? So tomorrow there is some disease which is dreadful, and we really want to discover a drug in a very short time. So then in that case, we really probably can do it. Now again, I'm not over promising, or I'm not misleading you by saying that everything is possible. Believe me, it's not. There are a lot of things. Human body is very complex. It gives you surprises all the time. That's where I said God also doesn't know whether a molecule in clinical trial will actually become a drug. Nobody can predict, right? And this is where, but anyways, so this is very interesting paper. I'll not get into this particular thing. This is another very interesting topic, antibiotic discovery, and many of the renowned scientists sitting here would agree. We have already run out of antibiotics. Believe me, by 2040, 2045, if we do not have good antibiotics, you fall from bike, you get an infection, and none of the antibiotics would treat you. You will die because there is nothing that will work. None of the antibiotics that will work. So you have a scratch, there is an infection, and you will die of that infection. Do you see the urgency here? Do you think it is really a dreadful situation? I mean, I don't know if you somebody who is into hospital and maybe in the terminal stage have suffering from some kind of infection or some complication, and then there is a hospital acquired infection, Pseudomonas aeruginosa. None of the antibiotics would work. They'll do all the antibiotic sensitivity testing. None of it would work, and they'll say that vancomycin has been the last resort, but then vancomycin resistant strains have been found, right? And if you see last 40 years, new antibiotic has been discovered. All the ones that have come, they are all the derivatives of already known antibiotics, including penicillin, cephalosporin. You should all read WHO report on antibiotic discovery. They keep on revising the priority list pathogen. They'll keep shifting from priority level 1 to level 2 to 1. You see how dreadful the situation is. And I would urge to all the future biotechnologists, you should take this antibiotic discovery as one of the goals or one of the areas that you would want to pursue. Mankind or humankind needs it. Believe me, it's going to be very, very dreadful. And this is where there are a lot of efforts that are going on. And this is just one of the approaches, where deep learning approach to antibiotic discovery, where on the left, on the right, what you see is the conventional approach, where you screen hundreds of thousands of molecules, whereas on the left, these people have used AI-based approaches. They have screened a very large library of molecules, and based on that, they have identified a lead, and then a lead as in a molecule which can go in human trials eventually, meaning after animal trials. Now, see, there is another issue that is happening. FDA is banning the use of animals in biomedical research. So, what will happen if you don't use animals in biomedical research? Now you are very, I don't know if you really use animals here, but then, if at all you are in some way associated with some animal research, if there are no animals, what are you going to do? As in, you probably have some surrogate models like organoid models, organ on chip, and then there are many other. You can't really take these molecules directly to humans. Meaning you can kidnap somebody, take them to an isolated place, inject them, whatever you would want. You can't do that. Nobody will actually. You need permissions. There are ethics committees, animal, institutional animal ethics committees. Even using animals in research, they will that committee members will try to reduce the number as small as possible, because animals also they also are living beings, and you just because you are so powerful, you can kill anybody, and then you take these animals for research. So there are a lot of issues that are happening, and this is where people are thinking how now. In fact, the regulators, those who approve the drug approval authorities, now they are also taking these things up in terms of accepting AI models as the substitution for the animals or even other approaches, other experimental approaches for that matter. This is again the same picture that I'm showing. So now this particular molecule, like I said, the target is new. We did not know when we started working on the phytochemical target, we did not know what target it was actually hitting. We had some idea, but eventually we realized that when we modified this phytochemical molecule, so then the need was arose that we wanted to understand what target it was hitting, and then we did formal target identification. So it was hitting a target which is located in smooth endoplasmic reticulum. Nobody has worked on that before, so it's a new target. There was the 3D structure was not available, so we went to 3 AlphaFold and told AlphaFold, see, this is our sequence. Why don't you tell me what the 3D structure looks like? And this is where AlphaFold gave me 3D structure, and I used for studying the binding of our molecules, whatever we have synthesized for with this target. So I've used AI, but then the original idea came from human mind, and this is where I would want to, again reiterate: AI is a tool. It is there to help. Not necessary that AI will replace all the human intelligence. This is where you need to understand its use. It's not a replacement for human. It is just an assist. It has an assistive role, and you should use human mind for doing more creative things and put all the redundant things or repetitive tasks to AI. This is a very, very important thing that one has to understand. This is another molecule that we worked on for some other target. Again, the 3D structure was we have developed using AlphaFold. These are some of the other things where now when I say lock and key, so a key has particular indentations, and that's exactly how it will open the key. It will open the lock. So here what we have done is we have used some ligand-based approaches. So this was for Ebola. The molecule that you see is it binds to one of the Ebola proteins. So again, I don't know. In 2016, there was an Ebola outbreak. Some people from Africa. Ebola is there since last more than 45 years. Nobody bothered because it was residing in African nations. Nobody bothered. Who cares? An African dies of Ebola. I'm not worried. But the moment somebody dies in London, oh my God, Ebola is a global threat. So that's exactly how. So like it's a business. Though companies will only be interested if there is an opportunity. So in 2016, something like this happened. So then, the drug discovery scientist in me, I like, let's do some computational work. So we undertook computational repositioning exercise using computational tools and already approved drugs. So we came up with a set of molecules which we said with the help of computational approaches, these molecules are likely to bind to Ebola and show the anti-Ebola activity. I had no access to any anti-Ebola testing or anything. So the editor of the journal said, well, these are just computational exercises. How do I believe this? But at the same time, some gentleman somewhere submitted a collection of 1,000 already approved drugs for Ebola testing. And fortunately, some of the hits that they have got and my hits from the computational study, our hits, so there was an overlap, and I could convince the editor, see, now there is an experimental evidence, even though we haven't done it. I'm not saying, I mean, we have just done this computational exercise, and that's how the paper got published. Some very, very interesting stuff that we have done. Again, AI is involved here. So this is my own paper published in 2016 for that matter. This is another very interesting study that we published in 2017. Molecular docking prediction and in vitro studies elucidate anti-cancer activity of phytoestrogens. Now, phytoestrogens basically from soya plant, basically, or soya oil, and then others. So this girl, PhD girl from NMIMS, so she came to me. I was working in NMIMS back then. So she came to me and she's like, the editor is asking. So again, the objective was for publication. She said, I have got fantastic results from the experiments, but then the editor is asking, tell us where these molecules are acting and all. So there were 4 of these phytoestrogens. So what I did is I again did reverse engineering in terms of where these molecules are likely to bind. So we did a thorough computational exercise, and then we sent the paper. And then the editor said, okay, now you are saying that these are the potential targets. Why don't you show it at the genetic level? So then the girl went on doing all the genetic stuff. I don't follow much of that. I'm a chemist. Pardon me for that. But then, so then again, she said, then they said, why don't you also show at the proteomic level? But then this girl, Sri Lekha Dutt, she was very determined, and then she did all the study. Very interesting paper, open access paper. Please read in case you are interested. So again, part of it was AI, part of it was human mind, of course. Right, so I'll just skip this again. This is from the same paper. When it comes to estrogens, the female sex hormone estradiol. So what you see here is in the pink is estradiol, and then what I have done, I have simply done the superposition of these phytoestrogens, and the shape actually matches. Remember the lock and key, right? A particular shape and particular aspect, and this is exactly where we were able to convince the editor that these were indeed the targets of those phytoestrogens. Amazing paper. I'm very proud of what we could do back then. This is another thing which actually we got this out along with Earth software. So 30th October on my son's birthday, we published this paper on KM Archive. AI designed family of dual COX-2 MP microsomal PGES1 inhibitors to treat inflammation with greater precision and effectiveness. Now this is on KM Archive, free paper. Amazing paper. We spent all Diwali days, 21 days, right from we decided to work on this. 21 days, full Diwali days in completing the work. Arshit, who couldn't come here, who is the brain behind all the AI stuff. In fact, the plan was to show demonstration, but unfortunately, he couldn't come. I would suggest you please go through this paper. Amazing work that we have done. We are very proud of. So right from the beginning, so he had generated few billion molecules, around 2.6 billion molecules using this reinforcement learning molecular generator. And then he has used multiple AI tools to screen these molecules for the docking, for the mutual fit, for the ADME properties, and a lot of stuff we have done. And then we have proposed a series of around 12 molecules, which we named as MAI, Mittal Arshit, and discovered by AI. So MAI, MAI 1, MAI 2, MAI 3, up to MAI 12. Amazing paper. There are a few structures we have shown here. But before we publish this paper, we actually filed a provisional application on Diwali day. We wanted it to happen on Diwali day. We could actually do it. Now we have 1 year to make these molecules and experimentally prove. Now, whatever we are, again, very important thing I would want to say, AI is giving us some predictions, but unless I experimentally validate it, it has no meaning. Don't think that AI is answer for everything. It's not. It will just assist you to generate hypothesis, and then you experimentally validate that. You actually do experimentation, but with the help of AI, the experimentation is going to reduce a lot, and at the same time, the cost is going to go down, and at the same time, the time which is required for to get to where you would want to go is going to be very, very less. Very, very fast. You will be able to go. Now, another interesting thing I would take another. Probably, I will rush through. I don't know how much time I have got. Okay, it's 12:40. So hopefully by 1 o'clock, I should be another 12 minutes. I'll be able to finish this. So biologics, I mean, biotechnologists would probably appreciate this. Biologics is the new wave. They say that 20th century was century of small molecules, and this century is going to be century of biologics. All the antibody drugs, all the nucleic acid based drugs, mRNA, siRNA, and whatnot, right? So, for you all biotechnologies, biologics is the area where you should be further, you should make a career in biologics. There are a lot of bright companies, startups who are working on multiple aspects of biologics. So I would want to bring to your notice, I don't know if you have heard about aptamers. Have you heard about aptamers? Anyone? Okay, so these basically are single stranded oligonucleotides, could be 40 nucleotides, 51 nucleotides, anything. And then you all know how nucleotides will. You have got a single stranded, typically DNA like deoxyribonucleotides. So the moment you have got a sequence, then depending on its sequence, it will fold in particular shape, because the complementary bases will actually form hydrogen bonds. So it will adopt a particular shape. So the idea is that these nucleotides, now you see, making nucleotides is very, very, it's not easy, but we have got oligosynthesizers. Believe me, I don't want to undermine. There are a lot of biologists here. I'm a chemist, but believe me, biology is very, very difficult, and I really appreciate all the biologists. Without biologists, we wouldn't exist actually. But anyways, so you can make these oligos. You can give the oligosynthesizer. This is my sequence, and make it in whatever time, 1 day, 2 day, whatever, small amount. I take these aptamers, and then these aptamers will bind to protein targets, and then they will inhibit, right? So making aptamers relatively easy because you have oligosynthesizers, and then using these aptamers for particular testing. Say like a lot of aptamers were reported for inhibiting the interaction of SARS-CoV-2 with ACE2 receptor. Amazing stuff. In fact, I did not know about aptamers. A friend of mine, he told me about it. So now, when I say aptamers, so first thing you have to do is you have to bring its 2D structure and then to 3D structure. Till like 6 months ago, predicting the DNA structure was very difficult, meaning RNA structure, RNA fold, and there are many, many tools. But it's just now with I think in last 6 months or maybe a little more than that, it's possible to predict the DNA structure. In fact, the first time I started working on aptamer, so it was a DNA aptamer. So what I did was I built using RNA composer, I built the RNA structure, the corresponding RNA structure, and then manually changed all the like two hydroxynucleotides to two deoxynucleotides, and then thymidine were replaced with uracil. I removed one methyl group so that the thymine becomes uracil and all. So very interesting stuff. So there is a process by which you can actually discover these aptamers. So it's called SELEX, but computationally also you can do it. And this is where there is computational SELEX. We are actually, one of my students is working on that. So basically you have a sequence, and then you come to 2D structure, 3D structure, and then you just do the docking. So there are tools available, AI-based tools available like Haddock, Plus Pro. You potentially can dock an aptamer, an oligonucleotide, folded 3D nucleotide to the protein target site, and then find out whether this is going to bind or not. Amazing stuff. So if you've not heard about aptamers, and if you really want to know more about it, so this is one case study from the literature, where I am sure you know about aflatoxin. It's a very potent toxin, basically, fungal toxin. Yeah. So what they have done is they have devised an aptamer who is able to bind aflatoxin. So just to it's like a determination or the determination of concentration of aflatoxin in whatever matrix it is there. So they have come up with an aptamer which will selectively bind to aflatoxin, right? So it's very amazing case study. Again, like I said, I can't really get into details, but they have designed the aptamer, predicted its 3D structure, and with the help of AI, they actually did molecular docking and found out how this aflatoxin was able to bind to this. Another very interesting thing is vaccine design, computational vaccine design. I am sure you have studied vaccines in great detail or you are going to study. So vaccine design is something which we are doing right now. The one which I am going to talk about in brief is a a pentavalent vaccine for M tuberculosis. That's another dreadful disease, right? Although it's India has a target of eradicating TB by 2030. I don't think WHO is 2035. India is very ambitious. Probably we may not be able to achieve that 2030 target. 2035 maybe. I'm not sure. A lot of things are happening because my sister is part of National Tuberculosis Program in some rural area. So she keeps telling me what is happening, and then this is really amazing without you even knowing what is actually happening at the grassroots level. But anyway, so with the help of a computational workflow, I will not get into the details. So we have designed a multi-epitope vaccine. So we have taken the antigenic peptides from the M tuberculosis, and then we have put them put them together in this using this workflow. And then the next thing was, so we have a sequence of around 299 amino acids. So what to do with that? So the next thing we did was we predicted its 3D structure using AI. And then we refined this particular thing, then identified the B cell epitopes, and although there is a computational workflow, ultimately now this is a vaccine, it has to activate, say, the antibody mediated immunity, right? Humoral immunity. So in that case, what we wanted to do was whether this is able to interact with TLR3, toll-like receptor 3. So we did the computational docking of the 3D structure of this pentavalent vaccine with TLR3, and with the help of this, we could show that the vaccine was able to do everything, like it was able to bind to TLR3 receptor. And there is one step going ahead. So there is computational cloning experiment that were also done with the help of this computation, everything based on AI, everything using free tools. In fact, currently we are working on glioblastoma vaccine, which is another very dreadful disease. Once somebody is identified of having glioblastoma, no matter what you do, typically in 15 months, that person will die. It's very dreadful. You read about glioblastoma. So what we are doing currently, or in fact, we are done, and this paper should be out by the end of this month on bioRxiv. So we have designed a glioblastoma vaccine, again, looking at the signatures of glioblastoma, the cancer. So we have designed a multi-epitope vaccine, anti-cancer vaccine. And then my dream is to really take this forward. So I have discussed this with one of the colleagues who is capable of doing this. So he's a molecular biologist. So we are going to really try hard. The only question is getting money, but we will see. At least we'll start with the initial experiment. So one of my students is working. So very interesting thing. This is the 3D structure of that MTB vaccine. And then this is the interaction of TLR3. That arc-like structure is TLR3. And then this molecule, the vaccine candidate, is binding with that. Very interesting thing, everything done by thing. Now I will take you to the last part, another 3 or 4 slides, and I'll be done. AI and computational modeling in diagnostics. Now, again, I am not a diagnostic expert, but whatever little I know, I have tried to pull this through from my understanding of medicinal chemistry, pharmacology, and then the uses of AI. Of course, image analysis has been at the forefront of use of AI. Now, there are multiple cases where an expert, a radiologist, so you look at a mammogram and then you see whether there is a cancer. You have to decide whether there is a cancer or not. So it's possible to have, the human errors can happen. There may be misdiagnosis or so, but now we have got very powerful AI models which can very accurately tell you very accurately identify with subtle differences in the normal mammogram versus the one with a diseased mammogram. So the AI can give you predictions. But again, it's not, the radiologist is not going to get replaced. It's just that it will be an assistive technology which will increase the efficiency, which will increase the accuracy of the prediction to a greater extent. Now here, of course, like I said, image analysis is at the forefront. So AI can analyze medical images and signals to detect diseases early and accurately. So you have an image from some examination, and then AI, you just ask AI. Now you see, if you are in a mega city or a metro city like Mumbai, things are there. But if you're talking about somebody who is in interiors and then there is no access to high-end healthcare, so in that case, some of these tools can be potentially useful. In fact, in 2015, I attended one workshop on AI in IIM Bangalore. So they have designed a model to identify a particular disease just based on the leaf images. So the farmer who is in the interior of Karnataka, all he has to do is just take the picture of the image which he suspects of the leaf, which he suspects that there is a disease. And then this AI model will accurately analyze that image and immediately tell the farmer that this is this particular disease. And then this is what the combination of insecticides or whatever the treatment agrochemical that you need to, so powerful. The farmer doesn't have to rely on somebody who will tell, come examine and tell. This is the power of AI, right? And this is where I really want to feature this particular example. Now you see, when it comes to cardiac arrhythmia, disturbed rhythm, so it could be atrial fibrillation, it could be ventricular tachycardia, and then there are multiple things. It could be heart block. Now, the accurate diagnosis is very important. Suppose somebody is suffering from arrhythmia, he is taken to the hospital. That person is probably not in a condition to talk. And then accurate diagnosis is very, very important. And one important thing is all they'll do is they'll record the ECG. So if the cardiologist misdiagnose it, because see, the medicines for atrial fibrillation and those for ventricular tachycardia, they are different. Mostly they are different. And then some of these medicines, they themselves are pro-arrhythmia, meaning they themselves can induce arrhythmia. So again, the point is accurate diagnosis and accurate treatment in very short span of time is very, very important. And this is exactly where AI can help. So all you have to do is, so you have an ECG, just scan. Of course, you have a backend model for that. So I could find this company infobionic.ai's MOMEARC. This is a proprietary model. All you have to do is just take a picture of that, ask the model, predict what it is, and it will accurately predict what exactly that type of cardiac arrhythmia is, which is, in times where there are emergencies, these things can be potentially very, very useful. Fantastic application. This is another thing related to CT and the MRI images, CT scan and MRI scans. So AI helps radiologists analyze CT and these images with higher accuracy and speed. So these models can detect tumors, stroke lesions, organ abnormalities, infections, and inflammations. Now you see somebody who's suffering from stroke, there is only a particular time, I don't know, 60 minutes or 90 minutes. Within that golden period, if you don't take the person to the right place, right medical facility, and if the person is not treated, the part of the brain will start dying. And this is exactly where, again, time is the essence. And this is where these images can help you at times, particularly for detecting tumors and all. So using an AI algorithm with non-contrast head CT scan that help clinicians better identify, localize, and characterize intracranial hemorrhages, which is it's like hemorrhagic stroke rather than ischemic stroke for that matter. So when it comes to AI in diagnostics and beyond images, so genomics and sequencing, it can identify mutations, it can predict disease risk. I told you, if you want to know whether in next 15 years you will be having cancer, your genomic signatures can be identified by AI. Digital pathology, so you analyze tissue slides using microscopy, using AI, and it will very accurately tell you what is happening at the molecular level. Wearable sensors and health monitoring, heart rate, oxygen, sleep patterns, stress levels. Now you have got sensors, you put it on your arm and it will keep on telling you every 5 minutes, it will take the, your blood sugar level. All you have to do is take your phone next to that and it will immediately show what is your blood sugar level. So you eat a samosa, it will tell you how much your blood sugar spiked. It will tell you every single thing. This is the power of AI. Amazing. That is even unheard of. Otherwise, if somebody is type 1 diabetic, you will not understand their plea, or you will not understand their situation, because before every meal they have to pray, find out what is the blood sugar level, or even type 2 diabetics for that matter, and then decide what dose of insulin is to be taken, and then inject insulin 1 month here, 1 month here, 1 month here, and then you keep on changing. Right. So, and then wound healing is another serious issue with diabetics. So you keep on pricking your body, and then wound healing becomes a problem. So likewise, you also have predictive analytics, forecast risk of diabetes, heart attack, or kidney failure using patient data. Right. So a lot of amazing things in case of diagnostic. So that brings us to the last part: challenges, opportunities, and lessons learned. Challenges: data generation and data quality. You feed the junk, and AI will give you junk, or ML will give you junk. Computational tools. So you need to have good computational tools, their capabilities and limitations. You should be aware of these. Computational resources. Again, I told you high-end computational tools, meaning the power is important. Then domain expertise, the domain expertise, AI is just an assistive tool. Domain expertise is very, very important. It's multidisciplinary and collaborative efforts. Like I'm collaborating with Earth, with Arshit to do things. I mean, sometimes I don't understand what he says in terms of hardcore AI. And many times when I tell him, he doesn't understand because it's hardcore chemistry. But we are still talking. It's like one person is talking, the other person cannot hear, and the second person cannot talk, meaning cannot speak. So likewise, we are just communicating. Right. And then successful integration of workflows in domain applications is very, very important. Opportunities: reduce experimentation, systematization of experimental work, increase throughput and quality output, expansion of applicability domain, and then study of complex and multimodal scientific problems, time, cost, and productivity benefits. Really amazing stuff that we are doing. Last year, I was in US. I had the opportunity to go to Merck Research Center, and then they are talking about spatial transcriptomics. So there are only two machines. One is in MIT, and these guys, they're just next to MIT in the Kendall Square Merck R&D center. And the second machine is there. So he was telling, so all we do is we take the clinical specimens, take the histology, and then ask this particular machine to do something called a spatial transcriptomics. So as you go away from your center of tumor, away from that, so it will tell you what is actually happening at the molecular level. And each analysis would cost around $5,000. Each slide will cost, analysis would cost $5,000, and it will generate massive amounts of data. And all you have to do is ask the AI to analyze that data to make some meaningful conclusions. Now, oncology trials are very, very deceptive, very tricky, right? So unless you have the right patient, the drug will not show efficacy. And this is exactly where AI is now giving you all that, strength. Yeah, seriously. I mean, it's really amazing, mind-boggling. So lessons learned: significant potential offered by computational tools. Change in mindset is required to move from age-old to new age approaches. In fact, yesterday we had a call with one of the companies, and all these guys, all the chemists from the chemistry manufacturing services or contract manufacturing services, all of them, they're like, they're used to think from a regular conventional approach. And when we said that these are the AI tools, and many of them, they were like, well, we don't think these will be useful, these, that, a lot of things happen. But anyways, that change in mindset is very important. Again, you have to be very realistic, and not that AI will do everything. Absolutely not. AI is not substitution for experimentation. It can reduce experimentation, but it cannot substitute the experiments. Models are dynamic and context dependent. Experimental validation is the key. So ultimately, you have to prove things by experiment, not just talking in the air. AI is doing this, AI is doing that, AI has given me this. But unless you prove it experimental, it has no meaning. Collaborative association is must for solving scientific problems. I mean, you cannot do everything all by yourself. You really have to depend on other domain experts. And this is where we have to let go our egos and then come together, talk to people, no matter how small that person is or how big that person is. Right. So patient is the most critical driving factor for delving into uncharted territories. I'll just, the last thing I would mention before I close, almost like 110, not a problem. So currently we are into one clinical trial. I'm helping like my team, and then there are other collaborators. We are helping a team of Greek physicians, neurologists. In fact, one of them is here in Mumbai. So they have come up with a I would say, very unique treatment option for glioblastoma. So after now, see, glioblastoma is a CNS cancer. After the cancer is scooped, it's very tricky. In fact, when you take out the CNS cancer, because if you touch the some part of brain which is involved in regular function, so you can be paralyzed, you can lose your speech, you can lose your eyesight, anything can happen. So they're always very careful when it comes to taking the cancer out. That means there will always be some cancer which will be left, right? And this is where these guys, they have come up with a combination of solutions, and then this solution will get inside. So they will insert the catheters, and then this solution will just keep on washing for 5 days at a particular rate. And they have already done compassionate trials where they have proved that this particular combination works. But now they want to do it formally, and this is where we are now getting ready for phase 1-2 trial. I'm helping them in getting the drug manufactured. It's already done, formulation done, and then we'll write to CDSCO, the regulatory body, for asking for permission. These trials are going to be held in Bangalore, Brains Hospital, and around 27 patients are expected to be enrolled, and then this study is going to. During this compassionate use, so both the patients survived. So one patient is alive since last 7.5 years. The other patient is alive since last 2.5 years, where the possibility is only 15 months. You see, now it's working. They're very excited, and I'm also very excited to be part of the cause, because ultimately, what do you want? I mean, how much money you have in your bank account, does it matter when you die? Absolutely not. But people will remember you for whatever you have done for the society, for this particular world. So you leave your legacy behind. Like we keep talking about all the Sakis, like last 30-40 years, they have been working. Now they have been working out of their interest for the society. Is it there any personal benefit? Absolutely not. Right. So this is exactly where you would want to leave behind the legacy. And I think this is what we are here for. Whenever we try to search why are we here on this planet, 75 billion people have come and gone. How many of them we remember? So are you going to be the next one to be remembered for something that you are going to do, or you are doing, or you will do? Right. So with this thought, I will thank you all for patient listening. And I'm very, very happy. No idea how happy I am talking about this. Thank you so much. If you have any questions, I'll